that paper is assignment is difficult, you better read the second paper again, especially the how to detect the gene escape. Okay, right. So now let's look at this our course today, the last section, successful stories of genetic modification. So far in plants, people have created so far cornworm or European corn borer resistant corns, bananas which produce vaccines, antibiotics, abiotic stress tolerant plants, onions that doesn't make you cry, onions that do not make you cry. That means they silence the gene of the chemical that makes you cry. Okay, golden rice. We talk about this. You read the paper about golden rice. Purple tomatoes. Why purple tomato? Because it's rich in anthocyanin and healthy, apparently. Carrot that help prevent osteoporosis. Okay. Soya bean oil for frying. Why frying? Because frying needs typical particular characteristics, right? So soybean oil, they have modified to make it for frying. And Arctic apple. Arctic apple is apple. When when you cut the apple, it gets the phenolic compounds, make it brown color, right? Now this apple doesn't have this oxidation process, so you can store cut open apple for quite a long time without applying anything, right? Otherwise, what you should do is citric acid or something you have to apply so that it will not turn into brown. But now this is naturally, it is not turning into brown, so that you can store, open it and cut and sell it as a fruit salad quite a long time. I mean, at least for a considerable period of time, right? Not like for years, but at least few hours, huh? Right. So another story that we are going to talk is blue rose story. They have created the blue color rose. So I know you and me, we are average people. We really don't care whether someone will give us a blue color rose or red color rose. Really doesn't matter. We like if they give us an apple, right? Or something edible, a chocolate, right? That is how we think. But people in like very rich people, very I should say very elite name is the wrong word. So very rich people who has nothing to do with money, they do care what kind of roses my fiance gives me. Okay, so this is a creation for that particular segment of the society, especially for those the wives of the kings of who live in Saudi Arabia or oil-rich countries. They are like, so that a real story is like that. Actually, the first batch of blue color roses was sold to these I can't remember the country, but wherever this oil-rich country, king of oil-rich country, so that he can gift it as to his fiance or wife or whoever, right? They earn quite a lot of money out of this production. Actually, it's a 10 years worth of research done by Japanese research group. After 10 years of continuous attempt, they generated the blue color rose. Not this blue, but it's a blue hue. They say blue hue. This is what they generate anyway. So you may feel like after seeing this, is this blue? And there are plenty of pretty roses out there. But I mean, I don't know. This is the first so-called flower. Okay, so the problem why blue color roses are really important. Why it is a matter? Why it is a news? The blue color flowers are everywhere, ne? Why it has become a news that blue color roses? Anybody? Any guesses from the class?
Naturally, there are no blue color roses.
Very true, Prabodha. There is no natural blue color roses in nature. Not only blue color roses, not many flowers are blue color. Yeah, Ambalika?
Madam, can you hear me?
Yes, I can hear you.
Madam, maybe there is no natural blue color roses.
Exactly, it's not because it's not naturally occurring. Very true. Not only roses. What are you going to say, Ambalika? Is that what you want to say?
Yes, madam. That means
yeah. Yeah, not only roses actually, even if you look around, chrysanthemum or lilies, many flowers, they don't have blue. In fact, in nature, only very few flowers are really, really blue, like butterfly pea, what we call katarolu, and some few plants that is shown here, like this. In nature, there are few plant species having blue color, like geraniums, uh, uh, certain flower types. So these are the ones that have natural blue color. Other most of the colors are in red, orange, yellow, white souls, region, red to yellow to orange to that coloration. Why this is because the natural blue color reside with the delphinidin pigment. Okay? So the delphinidin production is little costly. Maybe that is why these people who works in some priests like Devala, Kapua, and people, they ask you to bring blue color flowers for certain pujas, right? So maybe this is why. Who knows? Because it's rare, right? Blue color flowers, it's hard to find. So give an extra value, whatever. So Miss Joker, it's a joke, okay? Okay, right. So actually, most cultivated floricultural crops have been developed by a combination of extensive hybridization and mutation. For the mutation, so breeding, cross hybridization, when you don't have blue, there is no way you can get blue. Ne? So imagine you don't have blue color roses anywhere on earth. So by breeding, there is no way you can get blue color roses on earth. Therefore, people go for genetic engineering approach. That is the only approach we can go, or mutations. So, but making mutations to change the whole metabolic pathway is a lot of work. It's not really possible. So, the only way to achieve this is generating genetic engineering, right? So, before that, we need to understand the flower color or this color generation pathways, metabolic pathways. This is a kind of a metabolic engineering. Okay. So any engineering, genetic engineering, first we have to understand the metabolic pathway of the relevant genes, okay, of the relevant trait. In this case, since we are dealing with flower color, we have to look at the flavonoid biosynthetic pathway. Okay, so flower color is derived mainly from flavonoids, carotenoids, and beta-alanines. Okay, beta-alanines flavonoid, carotenoids, and beta-alanines. Okay, flavonoids and their colored class is mainly anthocyanin, which is anthocyanin, contribute to kale, yellow, orange, red, magenta, violet, blue. Okay. So it ranges. Carotenoids, we know it's a yellow color zone. Beta-alanines also yellow and red color. So let's look at the pathway. These are just extra information about flavonoids. Usually, where they store these flavonoids, these colors, in a cellular environment, where do they store them? They usually store in vacuole. Okay, vacuole, they store these colors, pigments, and mostly in glycosylated and acylated forms. So three major anthocyanidins are there: pelargoninin, cyanidin, and delphinidin. Pelargoninin, cyanidin, and delphinidin. Okay. When you have more hydroxyl group in the molecule, then it change into blue color shift. It shift into the blue color. Okay. So that is why we don't have blue. Naturally, classically breed roses, carnations, chrysanthemum, lilies, they do not have natural blue color pigment production. So therefore, all these species have to limit their color to white to red range, white, red, orange, yellow range, so on, right? Let's look at the flavonoid biosynthetic pathway. So here, this is the where the what I'm showing you is the and this general biosynthetic pathway of the flower color. You can see I don't want you to draw the structure unlike the herbicide resistance case. You don't have to know how to draw these structures. Huh? But if the pathway is given, you should be able to explain it. Okay, look at that. If I give a pathway like that, so you should be able to explain why plants, why they may not have blue color plants in that, like say for example, plumeria earlier or roses. Why don't they have blue color? Might be, look at that, to get blue color formation from the beginning of this point, you have to go extra length of the metabolic pathway, right? It has to go for extra mile. If it go one step, it will ended up in here, ended up in red color. Directly, it will ended up in yellow color or orange color, red color. To get the blue color, you have to walk extra mile. Okay? The pathway is little costly itself. Pathway you should be able to say, okay, that tells us why plants have minimum blue color because it's quite expensive pathway. So it's a long pathway to get into the blue color. What are the particular enzymes they have? Okay, one thing, these enzyme names you should be able to, like, at least you should be able to tell, like, okay, so F3H, F3H, F35H. So these are the two different enzymes. If you have all these things, and yes, then can go from either pathway. F3H in the media, if you have F3H, yes, it will end up in here. In addition to if you have F35H enzyme, yes, then it will decide to go for this pathway. Right? So it's the key is that the deficiency of F35H enzyme. When the plants do not have F35H enzyme like that, there is no way you can get into this pathway. So it's very clear that plants who do not produce blue color flowers or blue color fruits, they do not have this particular enzyme, particularly F35H enzyme is absent. So lack of delphinidine-based anthocyanins attributed to the deficiency of this particular enzyme. Therefore, naturally, there is no way you can generate blue color flowers because this enzyme is not there in natural varieties. So, since we do know that, so basically what we know now is F3-5-H enzyme or F35H enzyme, what we call. If you need the longer names, it should have been written here. There we go. These are the longer names. I don't much worry. Basically, these are hydrolases. Okay, so flavonoid hydrolases. So these hydrolases all are actually hydrolases. So these are all hydrolases. So in this case, it is hydrolase. So this hydrolases, this particular hydrolase is absent there. In carnation, this scientist research group found that okay, they wanted to create a blue color carnation, and they found that just supplying F35H enzyme to the white color carnations was enough to get the blue color carnation. And they found this is their years of experiments, because we know metabolic pathway. They know that in carnation also F35 hydrolases is not there, flavanol hydrolases is not there. So they overexpress it. They overexpress the enzyme, and that was fully, so they found that in carnation, the overexpression of this particular gene alone was insufficient to convert the metabolic flux fully towards the delphinidine biosynthetic pathway. So what they say is, if you overexpress this particular gene, or you just clone this particular gene from another, or you introduce this particular gene and let it express, that itself is not enough to get the blue color carnation. Okay. So they found that they tried, yes, there was a blue color. They got it like little blue color, but to get the real blue color, it was not alone. Expression of this enzyme alone was not sufficient. Okay. So they found that it's because the white carnation doesn't have DFR gene. Okay. So what they did was they found that they isolated, they found white color carnations, and it was the white color carnations doesn't have DFR gene. What is DFR gene? This one. They found white color carnation is there, and where white carnations lack DFR were transformed with petunia, this particular gene, in combination with DFR gene of petunia, generated the very bright colored violet kind of, like bluish violet kind of a carnation. What do they tell that? So what does that mean? So, okay, if you are going to make a genetically modified color producing plant species, first you have to study the metabolic pathway, see what genes are missing, what genes are there, and you have to try with one thing. Very first thing, it's very obvious if you and me, the researcher, we will try overexpressing this gene. So they choose the plant called petunia, like this. They choose this beautiful petunia flower, purple color, isolated the gene. Okay, F35 hydrolase gene was isolated. You can synthesize it, ne? And then insert into the carnation. It just inserting this particular gene only was insufficient. They found not much variation. When they insert into the white color carnation, they found it is because they don't have enough DFR gene. Internal DFR is not enough. See, because this is a metabolic pathway. Ne? So, plant tend to go for the easiest pathway. Why wasting energy? Even though it is there, plant tend to go for this pathway, and if you don't have enough internal DFR, this conversion may not be happen. If you don't have enough internal DFR, conversion is not going to complete. So what scientists did, not only this insertion, petunia DFR was also inserted into the white color carnation. White means no color. So it's easy. F35 enzyme and petunia DFR gene. And they found that accumulated delphinidine-based anthocyanin was almost exclusively generated, perfectly generated. But the same activity was failed. Now what researchers usually do, okay, you do one thing, you get carnation, blue color carnation, got the money from the market, and it's high selling, and it's like from all the flower exhibitions, they won the prizes and all. But then you want to try it for another plant species, right? So they decided to do it for the roses, because roses carnations are high priced, I mean, highly sellable flowers. So they tried with roses. They found that lack of blue color is due to lack of this hydroxylase enzyme, flavonoid hydroxylase enzyme. And they again decided to go for delphinidine production pathway. Okay. They found that they tried single enzyme, they overexpressed, and it didn't, they found that it's okay, overexpression of this particular gene was not any harm to the plant, because plant, you know, when you try to express a particular gene in a plant that is not there, if it is not there, you have to be very careful, because all the plant metabolic pathway we are shifting, ne? We are shifting the whole metabolic pathway. Maybe it is detrimental to the plant. Maybe it cannot tolerate this metabolic shift. So you have to first see whether this overexpression of this single enzyme is harmful or not to the plant. They found that it's not really harmful. Now that path is safe, even though the plant does not have this enzyme, you overexpress this, no harm to the plant. Plant happily survive. Okay. So then they tried like, there was a presence of pelargoninin and cyanidin-based anthocyanins derived from the endogenous pathway. So what are these? presence of pelargoninin and cyanidin-based anthocyanins. In the roses, pelargoninin, cyanidin-based anthocyanins are there in roses. Okay. Presence of these anthocyanins that are produced by the internal pathway prevented the coloration towards blue. It's obvious, ne? So look at that. So even though you supplement this one, even the any colored roses, they have these internal enzymes. They have the internal pathways. So having these internal genes, especially DFR genes, when the internal DFR is there, it tend to go for this, not with the foreign DNA. It try to go towards this pathways, not with this newly modified pathway. This is new to the plant, right? So plant tend to generate only red and orange color roses only. It prevent internal presence of these internal cyanidin and pelargoninin based anthocyanins prevented the color formation of blue. Again, the metabolic pathway shifting is not really efficient. Ne? So then they were trying so hard to find, is there any way, because it's because of the internal DFR gene actually. When you have internal DFR genes, your pathway goes the easiest pathway. Okay, even though it's introduced and it is there, working on this DFR, working on this particular enzyme was not happened in nature. DFR, you get this particular product, and when you have the DFR, it worked on this particular product, and theoretically it should produce blue color, but it didn't happen because internal DFR enzyme is there. So scientists search all the rose varieties to find any variety that doesn't have internal DFR. But they failed. There is no any rose variety that lack DFR activity. So all the roses they have had the DFR activity. Okay. Then what to do? So they had to remove or down-regulate internal rose DFR. They try to down-regulate rose DFR, up-regulate flavonoid 3,5-hydroxylase enzyme. They try to overexpress F3,5H gene, okay, obtained from viola plant viola. It's a blue color plant. They overexpress the F3,5H gene from viola, down-regulate the internal rose DFR gene. Now, see what they have done. Now, you try to get this particular gene from viola, okay? But still it doesn't work because internally there is DFR, rose DFR is there. So they try to reduce rose DFR. You increase the viola F3,5, internal DFR is reduced. Now plant doesn't have enough DFR, right? What you can do? They take DFR from another plant, Irish, another blue color plant. Irish DFR. They took Irish DFR, which naturally producing blue color. Okay. They took viola F3,5H enzyme, which is again natural blue color flower, supplemented this. Then they were able to stop these pathways because it down regulates. It's not working. Now the only pathway to do is this one. So scientists were able to get a beautiful blue color flower with the rose flower with the blue color hue. Is it clear? Is the story clear?
Yes, madam.
Anybody who thinks it's not clear or who thinks it's fascinating science, ne? Anything in there? Very beautiful vidya. It's a beautiful science. Sonali, don't you feel so? I mean, you should be able to feel like, wow, Didn't you get like wow moment?
Yes. Yes.
Okay, you really got it there. I don't know. So I think about individuals of yourself. Yes, Maheshika? Maheshi?
No, madam, I was trying to say it was very interesting stuff.
Okay. Yeah, so this is like really cool stuff. So how did scientists started the experiment? That is theoretical part. What if we do this thing? So what did scientists do really? They took several hundreds of rose cultivars. They screened initially by visual observations, very dark color, and red color cultivars were not chosen, not selected, because it contains too much of flavonoid and pH is too acidic. So even the color is there, it may not show up, right? So they remove all the dark color and uh, red color cultivars. Yellow and white cultivars were also not selected. Yellow and white cultivars were also not selected as they are not able to accumulate anthocyanins in petals. So the finding these things is another research project. Like these are very good master's research project. One student study all the rose cultivars on earth and see their pH in the flower in the petals. That's one project. And check their color anthocyanin and what other pigments available. Right? So they selected 169 selected cultivars. Okay. They subjected to flavonoid analysis and pH measurements. They found particular interesting variety and tried to supplement the biosynthetic pathway to supplement the missing genes, basically. They tried supplementing missing genes. So I think make a karadilama or a balagani never they do is they have to do something called they have to construct a gene cassette. Gene cassette. CSSED. Cassette. You have to make the gene cassette or we call construct. That is an example of one construct they create. They insert this into a vector, plasmid vector. Okay. So this is the segment, left border, right border. When you see left border and right border, you can get what kind of genetic engineering technique they used, right? What kind of transformation technique they have used. When you see left border and right border. What kind of technology they must have used? What kind of gene transfer method they used? Gene gun or what kind of method? Is the question not clear? I'm asking, when you see the gene construct means all the elements of the transgenes that you are going to transfer to the plant. Okay, they have done several constructs. They have they have tried several constructs to see the effect. Okay, one construct is that everybody would do very first. Small, simplest construct. Viola, F35H enzyme, promoter, terminator, right border, left border, and antibiotic resistant gene or herbicide resistant gene, whatever. Okay? So this is the simplest construct they created, and it has a left border and right border. What does that tell us? What kind of transformation system they must have used? I get it. Okay, I got answers. Anybody? When you look at the construct, it says there's a left border and right border. Why do we need this left and right borders? For what kind of transformation we need these borders? Okay. There are answers I'm getting. You keep answering. I'm not going to give. Okay, I'm getting few answers. One person say particle bombardment using plasmid to transfer. Someone say vector mediated gene transfer Yes, there is a vector, but I'm asking what particular system? There's a particular thing. When you hear the term left border and right border, you should be remembering something. Okay, plasmid vector, only express plant only. Okay, so many. Okay, I'm getting answers. Keep writing, keep sending emails, or keep telling me answers. ප්‍රශ්න පැහැදිලිද ළමයි? Okay, something to express only in plant. Another answer is using plasmid. Okay. Okay, what kind of plasmid? Like, I need a particular answer. I need very specific answer than this. Dr. Harshani must have taught you this. Vitushan, any guesses? You don't have to answer. You just send me a chat if you are worried that others may know the answer. Others may hope. Okay, I got one answer correct. One person, Yumi Sakuntala, correct? Any answers? You have to think and answer. Okay, I got Chamika, correct answer. Vitushan, you have to think. Yes, Nuantika, correct? Rethink. I am saying there is a left border and right border. You should be able to tell me, like, they have used this particular method. Okay, Sonali, correct? Minali, correct? Hassan? Any answer to chat? Send us a chat. Viturshan, you are correct now. Yes, you are correct. It is a plasmid, but we need the correct answer. Not every plasmid need this left border and right border. Only this kind of a plasmid need left border and right border. All the other cases, yes, Gayani, correct. Hasan, you have to think again. Ishanka, okay, I don't know why you write wild type. You don't have to have wild type name in front, huh? Ishara and Ishanka. Anyway, so what this thing is, so I don't know if you remember agrobacterium mediated transformation. In that case, remember you have a TI plasmid. Okay, this is T region. It has a left border and right border. Scientists say whatever in the left border and right border, middle, whatever things demarcated by left border and right border can go and integrate into plant. Therefore, if you are going to use whatever it is, left border and right border has to be there because we are using agrobacterium mediated transformation. Left border and right border is there, so the bacterial vector will accept it. And this vector is the agrobacterium TI plasmid that can go and integrate into the plant. So that is why they use left border and right border. When you have left border and right border, we know that is agrobacterium mediated transformation because left border, right border, if these sequences are there, we don't care. They don't care what is in the middle. This whole section will go and integrate into the plant. What is integrated into the plant is determined by these borders. Whatever in the middle, you can put any crop in the middle. Bacterium doesn't know, it will go, the whole section will be integrated into the plant. That is what we wanted, right? So this one construct, it's the simplest construct. They did that and found it is not going to work. Then what they, same as in the case of carnation, they did the same thing. You have the viola FT5 gene in plus DFR gene also. Irish DFR gene. Use that one also. Again, they found it's not enough. This one and this one. And then they use another Taurinia gene. In presence of, in addition to viola, they use Taurinia gene also. All these three did not give enough blue. It didn't do much change. Every time they found that internal DFR affected. Internal DFR affects. So even though you add outside DFR, it's not going to work. So scientists came up with the novel strategy that is shown in this particular construct. Now study this construct. Tell me what are the components of this construct. I will give you 2 minutes to think, and I will put you into groups. Think in the groups and discuss what these constructs are. What this group group yeah. You just think like individually. I should be able to explain what is NPT, what is the E35S Pro, Irish DFR, D8, E35 Pro, Master, Noster, every component you should be able to explain me what these components are. I'll give you 2 minutes. Think and get ready to discuss, okay? From cauliflower mosaic virus promoter, it is from the cauliflower mosaic virus. They found that this virus has this promoter that is constitutively expressed. Every time it's continuously expressed, very good promoter. Because viruses are rapidly developing, so they need a very nicely expressing promoter. So they use this 35S promoter in many cases. See, E35. All the cases they have used 35S promoter. Okay? And Irish DFR, it's sure that is DFR gene from Irish. D8 terminator. What is terminator? TER. Sorry, TER is terminator. Okay, I said that. Right. So they have used D8 terminator, mass terminator, NOS terminator. These are different terminator sequences that you can synthesize and ask the company to send you. Okay? Terminators are there. Now, terminator is there, promoter is there, Irish gene is there. That is clear. Let's look at this one. Viola F35 gene is also there. For that also you have to have terminator, promoter and terminator. That is also clear. Right border, left border is also there. What is NPT? What is NPT? If you don't know, just Google it. Yeah, it's a selectable marker. Particular name, if you try to particularly name it. I'm getting there. ළමයි ඔයලා බලන්න. Search, don't just waste your time sitting here. It's an antibiotic resistant gene, right? Okay. So they have NPT resistant gene. Oh, neomycin, no, this resistant gene, right? Now that is clear. Look at this particular section. This is what we have to understand. Terminator, this one, this particular segment. What is this? What is this? And it says cross DFR gene. What this thing is? Can anyone explain why this opposite direction little, what this thing is? Terminator promoter is clear. What is this black color? Arrows in opposite direction. Yes, it is a responsible gene, that is true, but why they are in opposite direction? Why this particular thing is there? Why is this? Think, Lamai, think. I'm getting two chats. Internal DFR, select particular region for the gene, internal DFR gene. It's not internal DFR. Yeah, yeah, it is for internal DFR gene, you are correct. But why they are in opposite direction? What you can guess why they are in opposite direction? And it's this part. So this is, they wanted to create a double-stranded RNA molecule. How? You imagine that now this is a gene. This is a gene. When they transcript, it produces mRNA. Okay. When when the mRNA molecule is producing after termination, that this particular molecule should be mRNA. When this gene has a repeated inverted repeat sequence, mRNA molecule has a sequence here, inverted repeat sequence here. It tends to form a hairpin in the cellular environment because they are compatible to each other. When the mRNA molecule forms a hairpin, this hairpin double-stranded mRNAs in nature are, there is a particular scenario, they involve in denaturation of the indigenous DFR genes. They go and double-stranded RNA molecules will go and find and cut the other RNA molecules of the cellular environment. Using dicer element, we call post-transcriptional gene silencing. Do you remember a term like that, gene silencing? Dr. Harshini must have taught you this. Is it not really familiar? Gene silencing, haven't you heard something called gene silencing? Yes. So what happens is that only I'm sure you, yes, okay, I get many yeses, yeah. So that means what happens is in the cellular environment, when you have double-stranded RNA molecules, it will go and try to destroy similar RNA molecules, like what we call dicer element. There are like a complex and denature the internal. So what happens is finally your internal DFR mRNAs will be digested. So it will reduce, internal DFR will be reduced. You get Irish DFR, you get this Viola F3 enzyme. And finally, that pathway, instead of going this direction, because internal DFR is reduced, the pathway goes from this direction to produce purple color or blue color. Otherwise, it will produce yellow, red. both of the pathways will be terminated because of the reduction of internal DFR molecules, silencing of the internal DFR molecules. So see how creative these scientists are, right? So how much thinking is needed. You have to think and do. These are very expensive experiments. Think very critically. Okay, should happen and do, and it happened. Imagine how much happiness they must have obtained after doing this. So you think, do, and you get the expected results. That is the, it's for a scientist. This is something that that you really want to get, right? Anyway, so this is how what they found finally. Wild type is shown in one end, and the other mutation is in the other end. So they choose this one or this one, I can't remember. They choose that as the best to go for the market. All the others they tried several different varieties. To me, I would love to receive a flower like that actually, but apparently the kings of the oil, this very rich people like this color. I think maybe, yeah. Anyway, so that is the story of blue color actually, blue rose. See, white color, it didn't get much interesting results, right? So they got this. This is a very nice change. See, orange to here, shifting the. See how much metabolic flux has happened in the cellular environment to get this much of a color variation, especially from this to this and this to this. Even to get a blue color hue, it's a lot of work. Then they did the northern blotting and so many other analysis to confirm that the mRNA molecule is there. Okay, so look at that. So rose DFR, look, see, this is the northern blotting. So you can see the molecule has been cut into pieces. See, so this is intact wild type. This is with that particular construct. You can see the digestion of the internal DFR, and Irish DFR expressing nicely. Viola, this thing is expressing nicely. So they have confirmed that not only in the color, they have confirmed it in the gels as well. Okay, so these are again the things. And right, so that concludes the section on blue color roses. And only one little thing I would like to teach you is, though there's a tutorial here, by the way. This is a study question or study guide, what we call study guide or study question. You can search answers for these questions and get ready for the exam. Okay, what are the gene hunting strategies? We have talked about that. How researchers have used SSR markers for high throughput assay. Okay, remember that we were talking about multiplexing of SSR, addition of using the 3' strategy, and what are the benefits of this design. You have 3' strategy. I have to do only one little section on gene editing, and this is concluded, but I'm to discuss in the tutorial. Do you remember these things like gene hunting strategies? Remember that we were talking about at the very beginning of the class? How to find a gene. Okay, and we talk about that it is like finding a needle in a haystack, but people do that. Matakai neda? Gayane? Remember? No. Okay, I get message. Thank you. Okay, right. And then we talk about how researchers have, so one strategy of finding gene is looking at the QTL assays, so whole genome screening. To do the whole genome screening, one method you can go for whole genome sequencing, or you can go QTL mapping in a mega scale, or whole genome-wide association studies, GWAS studied. You have mega scale high throughput genotyping method. One high throughput genotyping method is SSR markers changing into high throughput assay. That we talk about. You have used 3' and how to use it in capillary electrophoresis. And then I'm asking, giving reasons, just identify whether the following statements following are considered as GMO or not. Because we had a very big section on what is a GMO. What is a GMO? How to define a GMO? Is it same as genetically alteration, or is it same or different? Square-shaped watermelons. Have you seen square-shaped watermelon? In the internet, you will see that Japanese people create watermelons in different shapes. How? They when the flowering stage and fruiting stage, they cover the young fruit with a particular plastic wrap having a particular shape, heart shape or whatever, 3-dimensional, and the fruit develop into that shape. Okay. So then, is it a GMO or not? Is it a GMO?
No.
No. Why not? Because genetic composition has not been changed. No foreign gene has been inserted via recombinant DNA technology. See, two things has to be there. To define a GMO, you have to have a new gene. New gene that is not found in nature in that species. New gene that is not found in that species or genera in nature. It should be transferred using recombinant DNA technology. That means cutting, pasting, right? Using ligase, restriction enzymes, all these are used recombinant DNA technology and gene gun, agrobacterium, whatever. Golden rice, is it a GMO or not?
GMO.
It's a GMO because they have inserted two genes that are not naturally present in rice. What are those two genes? You should be able to tell what these two genes are. If you read the paper very carefully, phytoin desaturase and the other enzyme, right? So there are 3 enzymes that is supplemented by 2 organisms. One is daffodil, another one is Erwinia, another bacterium, right? So the one bacterium and daffodil use this for this process. So it's a GMO. Polyploid strawberries. Strawberries are usually polyploid, meaning that they are actually octoploid, 8, 10. So is it a wild type is 2 in strawberry, what we eat is octoploid. So is it a GMO? No, because this is a natural event. Naturally, chromosome number has been doubled. We did not use any technique. Drought-resistant, high-yielding F1 rice seeds. You have F1 rice seeds. They are drought-resistant, high-yielding. Is it a GMO? Is it considered as a GMO? No, why not? I get an answer saying no, why not? Hybrid seeds are often produced, always hybrid seeds are produced by biparental crossing. You have 2 parents, you cross each other, get the F1. F1 you cannot get from genetic engineering. F1 is hybrid. Hybrid is you have to have a mother and a father, right? So they are not GMO. Of course, genetic composition has been altered, genetically altered, but it's not a GMO. Okay, comment on the following statement. Genetic transformation can happen only in nature. Is it true or not? Genetic transformation can happen in nature.
False.
Only in nature.
False.
It's false. Why it is false?
We can do it artificially.
Exactly. We do artificial genetic transformation. How do we do transformation? You have to cut open a plasmid, insert a gene, and seal it, and clone it. And that particular cloning process, you transfer your modified plasmid to the organism. So it's a altered organism, genetically transformed organism. And now we can do it artificially using what are the methods? You can do it using heat shock method, calcium chloride transformation. You can use viral vector, Or you can use electroporation, making artificial holes by electric shock. Matakai neda? Transformation techniques. Gene transfer techniques or gene gun, whatever, right? So this can happen in nature. How they know that it has happened in nature? By studying the genetic material, they have found horizontal gene transfer. They found horizontal gene transfer that some fungi obtain toxin genes from bacteria. So genes horizontally transfer across kingdoms. So it happens in nature. Not only in nature, now we can do it in artificial, in the laboratory also. Okay, recombinant screening and selection is two different things. Screening, selection. Library screening, library selection. If not, look at your old notes. And selection and screening is two different things. Where do we use selection and screening? Selection is when you have many things, you have to select what you want. Screening is you need to let the you read this. This is very clearly mentioned in the notes. Okay. Genomes can be expanded as well as shrink. Is it true or not? And give examples. Comment on this particular statement. Can genome be expanded over the evolution, over the course of the evolution?
Yes, ma'am.
Yes. What kind of mechanism of genome expansion? Insertions, repetitive elements, the pseudogenes, making many copies, gene duplications, genome duplications, and remember that when the chromosomes are not really separating during the meiosis. Okay, so I can't delete this. Anyway, so read about that. And genome can be shrinked. Genome shrinking, they lose genes. Genomes tend to lose genes. They have identified it in Mycobacterium tuberculosis case. Read your notes. It's clearly there. Okay, differentiate a genomic library from a cDNA library. Can you differentiate genomic library from a cDNA library? The differences between genomic library and cDNA library. Can you think of this? Genomic cDNA. What is the starting material for the genomic library? DNA, right? cDNA library, what is the starting material?
RNA.
RNA. Yeah, now you should be able to describe how to extract, how to do the cDNA library construction also. That's the next question. Which one is bigger, which one is smaller, what kind of vectors are used, what is the initial starting point, and which one is easy to screen. All these things you should be able to compare. List essential steps of constructing cDNA library. You get RNA, you convert into cDNA. Remember first strand synthesis, second strand synthesis, and the first strand, and then cDNA synthesis and digestion of the mRNA molecule, and use the S1 nuclease to digest the loops and make double-stranded cDNA and clone it, and selection and screening. Not really details like if you get blunt end ligation, no need to have this every single detail. Key steps of you should be able to do the key steps of the genomic cDNA library construction. Especially what I'm interested in is isolation of RNA, separation of mRNA. How do they separate mRNA using poly-A tail? Okay. And then synthesizing of the first strand and the second strand. S1 nucleus digestion, all these things has to be there. Okay. Study how to calculate the number of clones need to be screened to find a particular gene. We did this using two different methods, using an equation. You should be able to do the math. Quick make a karani. if an abstract is given, like in the case that I gave you an assignment abstract, a big abstract from the research paper will be given, you should be able to draw a construct. That we talk about constructs like this. By reading the abstract, you should be able to draw the construct that researchers might have generated. All the information will be given, and you should be able to draw a construct. Explain the CRISPR-Cas gene editing system. That is what I'm going to teach you now. And design an experiment to test the herbicide-resistant transgene explained to the environment. This question was from the last day, first assignment. Remember, I gave you a paper to read, Gene Escape. That this question is originated from that paper, and you should be able to answer key steps, not really in detail. If you are a researcher, and if farmers say that my fields have herbicide-resistant plants, weeds, and you suspect that it came from another wheat field, another herbicide-resistant transgenic experimental field, how you are going to prove it, how you are going to analyze it. Okay, so that concludes the section. Only thing that we are left is CRISPR-Cas gene editing system. Okay, how many of you have done CRISPR-Cas, learned CRISPR-Cas before? Raise your hand if you have heard about CRISPR-Cas gene editing system. One person. Only one? How many of you have not heard about CRISPR-Cas gene editing system, CRISPR-Cas? 1, 3, 4, 5, 6, 7, 8. Okay. I'm getting many numbers. Right. Let me explain what this thing is. So remember, CRISPR-Cas gene editing system, or whatever these gene editing systems are not, these are not genetic engineering technique. Is it clear? I don't have a note or slide for this. You take your note while you are listening to the video. Okay? You look at the video, take your own notes, and let's conclude it like that. Okay? Any questions so far? Before, up to this point, any questions? Ashna තියෙනවද? වේගෙ වැඩිද? Lamai, do you have any questions? Oh, okay. Is everything okay? Ambelika, is everything okay?
Yes,
Okay. Purnima?
yes, ma'am.
Okay, right. So let's learn about this CRISPR-Cas gene editing system. Remember, USDA, United States of Department of Agriculture, clearly states that CRISPR-Cas gene editing system is not a GMO. It's not a genetically modified organism made. It's not belongs to, if you have a plant that is made out of CRISPR-Cas, it's not a GMO. You will understand why it is not a GMO after learning what CRISPR-Cas is. Okay. So can you see my screen? My screen එක දැන් පේනවද YouTube එකක්? Hold on. Okay. Yeah, what I'm going to do is I will stop and I will explain it in simple English. You take your notes, okay? Okay, what does that mean? Every cell in our body, every cell contains a whole copy of our genome. Okay? Even the cell from eye, cell from hair, cell from nail, everywhere we have the whole genome. But how it makes a nail? Only those genes that are required to make a nail are expressed in the nail. Those out of these millions of the 30,000, 20,000 genes, those which are required only to make an eye or some kind of a cells in eye, they will express, only those genes will express in the eye. Same in the hair. In the hair, even though every cell has a 30,000 gene, not every gene is expressed. Those genes that are required to make a hair, those genes will be expressed, right? So I hope the difference is clear. Even though every cell has a whole complete set of genes, not every gene is expressed at the same time. Yeah.
And held together by a simple. It consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes.
So that is clear. Our genome is consist of double-stranded DNA, which is pairing by complementary pairs.
Shape who we are as individuals and as a species.
So our genome tells us who we are and our shape, morphology, physiology, everything.
Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease.
Okay, with the Human Genome Project, they have identified hundreds and thousands of genes that are responsible for diseases.
To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy.
Okay. Now, if we are a living system, changing our genes in the living system is not easy. Okay. While we are, it's inside the tube, it is true. Yes, you can change it. But while it's in the living system, it's not easy.
But recently, a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including.
Okay, but recent technology helped us edit the genome in the system. Okay, we can edit our genome. Editing means doing little minor changes like removing one nucleotide, adding another nucleotide, replacing a nucleotide, a few nucleotide changes, deleting a few nucleotides, something like that we can do now. How? Because of this particular natural system they have found.
Humans. The CRISPR method is based on a natural system used by bacteria to protect.
Okay, so this method is very first found in nature. Okay, that is found from bacteriophages. So bacteriophages are a kind of a virus that infect bacteria. We know that bacteriophages infect bacteria. So bacteria have their own mode of protection against bacteriophages. How they protect themselves from bacteriophages? Let's see.
Protect themselves from infection by viruses.
What that happens is when the bacteriophage infect the bacteria, it releases its genetic material to the bacterial cell.
When the bacterium detects the presence of virus DNA, it produces two types of short RNA.
Okay, so this is the interesting point. When the bacteriophage, or what we call virus, it's a virus, right? So when the bacteriophage releases its DNA to the bacteria feel that, okay, there's a new foreign DNA molecule. What I should do? They produce two types of short RNA molecules. They produce two types of short RNA molecules as soon as they detect this presence of this virus.
One of which contains a sequence that matches that of the invading virus.
Okay, so the beauty of this thing is one RNA molecule that is produced by the bacteria has a sequence that is similar to the invading RNA, invading molecule, invading viral nucleic acid. Okay, so that is same as the little section is similar to the invading molecule. So bacteria has a mechanism to copy and make a similar kind of a RNA molecule. Okay, so one molecule is matching to the invading virus.
These two RNAs form a complex with a protein called Cas9.
So one RNA molecule having the similar sequence to the invading virus and another RNA molecule. Two RNA molecule form a complex with the protein called Cas9. Okay, so that is called Cas9 complex.
Cas9 is a nuclease, a type of enzyme that...
So when the RNA molecules and the Cas9 is there together, it works. It's an enzyme. It's an endonuclease. Endonuclease means it can cut DNA. So this complex can cut DNA. What DNA is the question? Let's see.
Can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome.
Okay, so the RNA molecule that is produced by the bacteria, we call guide RNA. Why it is called guide? Because it can go and find and bind with the foreign, when you have a foreign visitor, we go and... Ane, thank you.
The Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short...
Sorry, I had a visitor, so I had to attend to that. So is it clear now? So up to this point, is it clear? Anyone who thinks needs my explanation? Raise your hand, please. Anyone who needs extra explanation? 1, 2. Okay, 2 members. Okay, 3, 4, 5. Okay, right. Okay. So from which point you need, you think it's not clear? Okay, let's go from this point. Okay, let's go from the beginning. Beginning as in a little bit beginning. Okay.
Thousands of genes that affect our risk of disease.
We have thousands of genes, that is clear, right?
To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy. But recently, a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection.
Okay, so this method is used by a natural system. Naturally, bacteria are protecting themselves. The bacteria protected themselves from the invading bacteriophages. Bacteriophages come and attack bacteria. So bacteriophages attack, the attack caused by bacteriophages is being prevented by a natural system that is present in bacteria. What that natural system is, they produce 2 kinds of RNA molecules. One RNA molecule is matching, complementary to the invading virus DNA. Okay?
By viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the virus...
Okay, what happens is, up to that point, is it clear? Is it clear up to that point, Vinuri?
Yes, madam.
Okay. Anybody?
Madam, that means
Yeah.
that RNA part is already present in the bacteria cells.
No, bacteria synthesize it when as soon as it's a process. So when the bacteriophage release its nucleic acid to the bacterial cytoplasm, it's a big process. They detect the severe changes. As soon as the bacteria sense that foreign DNA material is inside my cell, it synthesizes a short RNA piece that is complementary to the invading virus. In some cases, scientists found that it is already there because that naturally it can also it can be there as well. It's an inherited. I mean, it's a character. It's an inherited thing. Is it clear?
Yes, ma'am.
Yeah. So either way, somehow bacteria detects the presence of the virus. And it synthesizes two kinds of RNA molecule. One RNA molecule has a matching sequence to the upper. Okay, my computer is slow. Okay, that's another thing. Right, so it detects the foreign DNA molecule, and then it goes and bind with the matching RNA molecule, try to go and bind with the invading viral nucleic acid, like this.
Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus.
Look at that. It's beautifully cut the DNA molecule. So when the guide RNA goes and bind with the invading viral DNA molecule, and this is not, it doesn't go just RNA molecule, it goes as the Cas. Cas means Cas enzyme and two RNA molecules. This complex itself is a nucleus. It has the ability to digest DNA. So when this, how this guide RNA, so this is like a guide, a tourist guide, come and go and show the pathway, right? Likewise, this guide RNA help the Cas9 system to show the foreign invader. Okay, it goes and bind with the invading RNA molecule because the sequences are matching. As soon as it's bound, this whole system is the enzyme. It digests the foreign DNA, foreign nucleic acid molecule from the virus. So the virus will get digested.
Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location.
That is the interesting part. So So that is the interesting part that they are talking about. So it's not just the inside the, give me 1 second, Lamaya.
By changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM.
So that is the beauty. So in here, when the it's not just it can cut only virus RNA, any RNA molecule you can digest and do some little editing in nature. So scientists study this system for a long time and found that okay, this can be done in any system. So how it has been modified is like this.
The resulting complex will lock onto a short sequence known as the PAM. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA.
Okay, here what happens is in the living cell, you can really synthesize any nucleic acid sequence as you wish. If you like to change any particular, like in the case of bacteria, it synthesizes a similar sequence to the invading virus. Similarly, you can synthesize a particular short RNA molecule having a particular sequence known as PAM. Forget about the PAM, don't worry about PAM. Having a similar sequence, you can synthesize the RNA molecule having a similar sequence that you like to edit, so that it will go and bind and unzip the DNA, bind and edit it, right? And digest it. See how it happens, huh?
And match it to its target RNA.
You have the matching location.
If the match is complete,
It's match
the
complete.
Cas9 will use two tiny
Cas9
molecular scissors
digested.
to cut the DNA.
Now the DNA has
When
been
this happens-
digested. Look at that. Now, after opening, nucleotides are losing. Okay, after this digestion by this Cas9 system, the nucleotide, they lose some nucleotides from the cut point. Okay.
The cell tries to repair the cut.
Now, once it is cut, cell try to repair it. Okay. This repair can happen in two ways. Let's see.
But the repair process is error-prone, leading
This repair process, it tried to complete this gap. It tried to fill the gap. It tried to get some whatever the nucleotide and try to fill the gap. When it tried to fill the gap with whatever the nucleotide, it is an error-prone system. Now it used to have a G. Now you have thiamine, right? Something is an error-prone system. So this is really good if this is a cancer gene. If your gene is a cancer-causing gene, you try to make a mutation there. Now it no longer able to divide because it's just there's a change in the nucleotide order. It's no longer the same gene, right? Is it clear?
leading to mutations that can disable the gene, allowing
Now,
research.
when you have mutations, gene is disabled. Is it clear, Lamai? Beautiful, isn't it?
Yes.
Yeah, right. So basically, you can disable a gene like that. You just digest, cell itself try to repair. Of course, with problematic nucleotides, the gene will disable, it will not be active, which is good in terms of cancer genes, something like that, some disease-causing gene, right? Now, you can do the repair in another method as well. I don't know if they talk about this or let's say, yeah?
Researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise. For example, by replacing a mutant gene with a healthy copy.
Look at that. So that is a random process. Yes, some nucleotide will come and bind and gene will be inactivated. Yes, it's good for a devastating gene, it's good for a bad gene. But sometimes if you need to cure a disease, like you need to replace that particular gene with the correct nucleotide. Correcting. You have to do the little correction. You have to be very precise. Scientists say you can do that as well.
This can be done by adding another piece of DNA that carries the desired sequence.
You can add the desired sequence and insert it.
Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and...
Exactly. The nucleotide is cut, two DNA strands are there. This foreign DNA you insert with the correct nucleotide sequence, it comes and it makes a single strand, and using that single strand as the template, it makes the other strand complete. The main strand එකක් තියෙනවා, අනිත් strand එක synthesize කරගන්නයි තියෙන්නේ.
Replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types.
So the beauty is you can do this in the normal cells as well as in the stem cells, in the plates, petri dish, living tissue. So if you can do this in correction in the stem cell, stem cells, have you heard what the stem cells are? Can anyone explain what stem cells are? Stem cell research is a very big area in these days. Okay, I got one chat. Embryo. Stem cells are the cells that can regenerate into any organ. In the body, we have certain cells that can regenerate into any organ. You can make from that particular cell a full heart, or you can make a bone or whatever, right? So it's like a spare part factory. I mean, I'm just exaggerating, but the thing is like you can convert into any particular cell. They are located in inner cell passages of embryo or bone marrow or certain places. People use this stem cell research to replace as a cure for the cancers as well. Actually, one of my friends took this stem cell therapy. Of course, she lives not in here in Australia. She got the cancer, so she used this therapy, and apparently, stem cell therapy is like a leading thing in these days, huh? Right?
It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting.
You can do this in a human cell line. If you have a cell line, you can edit many genes like that and study the causes for the disease and what mutations cause the disease, everything. So it's a very advantageous system.
Together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
So the problem is not the problem actually. So the scientists very recently have found that actually there is another cancer risk as well with the I mean, there are things like that, you know, we for certain time we think, okay, CRISPR-Cas is the key and it's the best and all these things, and later on they found, okay, even when the cells are subjected to CRISPR-Cas as a unexpected cancers can occur, and there are things like that. There is no single silver bullet thing, but this is one fancy method that you can use for gene editing. Remember, gene editing is not a GMO because we are editing our own gene, not inserting. I mean, even if you insert that correction gene editing system, when you correct it, still it doesn't belong to GMO category. Okay, so that concludes the genetic engineering section for today. Any question before we take a break? Questions? Tutorial is very important and the assignments are very important for your exam. Anything that is not clear, anything that you wish if it is more explained or something? What you just completed is a very high quality program actually, course, because you kind of have covered everything required for the genetic engineering course. Everything kind of is covered, and depth of the knowledge you gain is lot. And I know you don't enjoy reading papers, but that exercise is also has been done thanks to the strike, the whole day exercise, which will help you to practice reading, because if I give that assignment to do alone, no group, nobody will read it. And this is like one day when you have a deadline, you have to do this. Ne? So it's a really good opportunity. Reading, Lamai, as master student, as postgraduate students, reading is the key. Not reading my notes, but reading the research papers. When you're especially doing your research, you should be able to read the paper, interpret it, and do it in your setting. Okay. Anyway, anyway. Yeah, Piyumi?
That madam, healthy DNA part is how we insert it as some method. How we
you mean in the CRISPR-Cas, right?
Madam?
In CRISPR-Cas, I say, right?
Yeah, madam.
Yeah. So what you can do is, because that you have to have the prior knowledge. With the previous research, you should know that, okay, this is the mutation of this particular gene. If that section is deleted and replaced with imagine in a normal cancer patient or any other patient, you have ATTT gene, ATTATTCG, and if the normal cells have GGGT or something, then you have to synthesize that GGGT section artificially. You can synthesize it artificially. And then you can send it to the cell. That part you have to know the previous knowledge and you have to synthesize artificially. You cannot let the cell synthesize inside. It's the artificially synthesized material, like an injection, right? You inject the RNA. corrected RNA or corrected DNA. Any other questions? Questions, guys? Prashna Tinwada? Did you enjoy the session? No?
Yes, madam.
Okay. Right. Any questions? Okay, so if so, we'll take a quick lunch break. Now it's 12:41, let's say it's 12:45. Let's come back by, is 15 minutes okay for the lunch? Quickly,